We're moving on now to the second lecture in the Streptococcus lecture, and we're going to start out by talking about the organism Streptococcus pyogenes. Streptococcus pyogenes is also group A Streptococcus because of those Landsfield antigens. You can see in this slide on the right the gram stain of Streptococcus. Now, unlike Staphylococcus that tends to cluster up like grapes, Streptococcus tends to chain together. So in clinical samples, you'll see nice long chained organisms, or in some cases paired organisms. However, strep can sometimes look a little clumpy too, so you must do at least a catalase test to differentiate your Streptococcus from your Staphylococcus organisms. This is Streptococcus pyogenes, grown on different type of media, just a general supportive media. Streptococcus pyogenes has M proteins that will allow for surface attachment to host cells. It can produce a capsule, which is antiphagocytic. It has very specific toxins and enzymes that it can produce, such as hemolysins, which will hemolyze red blood cells, deoxyribonucleases, which will degrade DNA, hyaluronidase, we already mentioned with Staphylococcus aureus, which is known as spreading factor that allows the organism to spread through the tissues, and it also can form an erythrogenic toxin, which will produce a rash in patients infected with strains that produce this toxin. Streptolysin O is produced by the group A streps. This is an oxygen labile virulence factor. It is more active deep in the colonies where that oxygen tension is low. So oxygen will kill Streptolysin O, so it likes to be in low oxygen potential. And this is why for clinical specimens, when you're in the clinical lab, if there is a sample that might contain a Streptococcus, you really should stab the quadrant, at least the first quadrant. Some, depending on what lab you work in and how you're trained in the laboratory, different labs will do different things. Some will put a stab in each quadrant, some will only stab a certain quadrant. So you want to stab the media in order to get the oxygen into that area and to get the organism down into that area as well. Streptolysin S is oxygen stable, so it's perfectly fine in the presence of oxygen. It's cell bound, and this virulence factor inhibits chemotaxis and phagocytosis and can injure cellular membranes. Erythrogenic toxin is a pyrogenic exotoxin produced by group A strep species, specific strains that carry a lysogenic phage. And remember, a phage is a virus that infects bacteria. So only those group A strep strains that have this phage will be able to produce the erythrogenic toxin. This toxin causes rash, and it's commonly found in patients with scarlet fever. Deoxyribonuclease, or DNAse, is an enzyme that degrades DNA. You can use anti-DNAse to diagnose acute glomerulonephritis in patients that are infected with the group A Streptococci. DNases degrade DNA that might accumulate in pus as well, and that might increase the spread of Streptococci from one localized area and go on to other areas. Hyaluronidase is the enzyme that's called spreading factor. It degrades hyaluronic acid in the connective tissue, and it facilitates spreading of organisms throughout the connective tissue. For the clinically significant streptococci, you have several structures. There are fimbriae that will allow for attachment of host cells and adherence, that M protein, the spreading factor, there can be capsules. So Streptococcus has the ability to produce numerous virulence factors. Group A streptococcal infections. Some of the infections include acute bacterial pharyngitis, so basically a throat infection. Patients with bacterial pharyngitis will have a sore throat, might be tired, maybe have a fever, slight headache. Group A strep, or Streptococcus pyogenes, also causes the disease scarlet fever. It also causes skin infections like Staphylococcus aureus. Streptococcus pyogenes causes impetigo. If you remember, that's that skin infection that's very common in children and is highly contagious. And it also causes a pyodermal infection called Erysipelas. So pharyngitis is most frequently found in children, caused by Streptococcus pyogenes, or group A strep. It's also known as strep throat. Causes an acute sore throat. Usually is self-limiting, however, many times antibiotics are needed. And the thing about Streptococcus pyogenes, or strep throat, it can spread and cause otitis media or ear infections, sinusitis. If it gets into the lungs, it can cause pneumonia. It can cause blood infection. So if it isn't self-limited or controlled through antibiotics, it can cause a much more severe infection. Impetigo, we already mentioned. Erysipelas is an infection where you'll see the area get very red, get swollen, be hot to the touch. And there's also puerperal infections, such as infections of the endometrium. This is a picture of impetigo, again, that skin infection that's highly contagious. And I already said it, but whenever we talk about an organism and a disease that it causes, you always have to think about what other organism causes impetigo. And you remember it's Staphylococcus aureus. For microbiology, it's very easy to memorize your notes in little sections, and to take the quizzes, that's perfectly fine. But keep in mind, when we have an exam, if the question are which of the following organisms can cause impetigo, you have to remember that Staphylococcus aureus can cause impetigo, but also Streptococcus pyogenes can cause impetigo. Strep throat is the most common infection, most well-known infection that Streptococcus pyogenes causes. Cellulitis, where you get that tissue infection, where you get a lot of swelling, redness, heat. It's also called erysipelas. And Streptococcus, like Staphylococcus aureus, can produce toxins. So Strep pyogenes is the cause of scarlet fever. It produces an erythrogenic toxin. You get a red rash on the skin, and you can get hemorrhaging on the palate of the mouth. Like Staphylococcus aureus, Streptococcus pyogenes causes a toxic shock-like syndrome due to a toxin called exotoxin A, which is very similar to Staph aureus's toxic shock syndrome toxin 1. Group A strep also causes the terrible disease necrotizing fasciitis, or the flesh-eating disease. Invasive group A strep infections include that Streptococcus toxic shock-like syndrome, which, just like Staphylococcus aureus's toxic shock syndrome, it causes multi-organ system failure, it leads to shock, and it commonly leads to a fatal infection. That necrotizing fasciitis, group A strep or Streptococcus pyogenes, is also called the flesh-eating bacteria. The media likes to call it the flesh-eating bacteria. So this is a severe infection. It starts out as a cellulitis. It then spreads, leading to a bacteremia and sepsis. It gets into the organs, and usually with these infections, amputations have to be done to try to eliminate that toxin from spreading into critical organs. And here we have some of our disgusting pictures. So for those of you that have a queasy stomach, you might want to fast forward. And here's some pictures of necrotizing fasciitis, where people have had to undergo amputations. Usually the area has to be debrided, that tissue has to be completely removed, and many times the toxin will spread too rapidly to major organs and the patient will die. Some of the non-suppurative sequelae that group A strep causes, acute rheumatic fever is one of them, where you have inflammation of the joints, the heart, and the central nervous system. This can last for 2 to 3 months, and individuals that have acute rheumatic fever can have a carditis that can lead to heart failure. Acute glomerulonephritis, or infection in the kidneys. You can see high blood pressure, you can see protein in the urine, blood in the urine, and this can lead to kidney or renal failure. Individuals that have group A strep infections can have some more chronic manifestations. So individuals, some children that end up getting a throat infection that leads to it can lead to rheumatic fever, and that can generate a chronic heart condition in these individuals. Individuals, again, with the acute glomerulonephritis can end up with chronic kidney disease and kidney problems for the rest of their life. For lab diagnosis of group A Streptococcus, you will see the nice gram-positive cocci in chains in a patient specimen. So here you can see a lot of white blood cells and the gram-positive cocci in chains, and this is a wound specimen. On blood agar, the organism is transparent, smooth. It has a well-defined zone of beta hemolysis, so that's critical. Streptococcus pyogenes, or group A strep, is beta hemolytic. Because it's a streptococcus, it is catalase negative. It's susceptible to bacitracin, so you make a lawn of the organism on an agar plate. You drop a disc that contains bacitracin in it. If the organism can grow right up against it, it's bacitracin resistant. If the organism cannot grow right up against the disc and there's a zone around it, it would be bacitracin susceptible. So group A strep would be shown on the left because there's a zone around the disc. Group A strep is also PYR positive, bile-esculin negative, and it cannot grow in the presence of 6.5% NaCl. We're going to move on to Streptococcus agalactiae. This is group B Streptococcus because of its Lancefield antigen. It is also, like Streptococcus pyogenes, it is a beta hemolytic Streptococcus organism. Group B strep can cause mastitis in cattle. It can colonize the urogenital tract of pregnant women, and it can cause an invasive disease in newborns, and this is due to passage of the baby through the birth canal in individuals that are carrying group B strep in their urogenital tract. It can cause an early onset infection in newborns or a later onset disease. In infants, Streptococcus agalactiae is the leading cause of pneumonia, sepsis, and meningitis during the first 2 months of life, and this is usually due to delivery, contamination during delivery from the urogenital tract to the newborn baby. In children and adults, group B strep can cause puerperal fever. It can cause respiratory infections, meningitis, bacteremia, endocarditis, and also urinary tract infections. Some of the invasive infections caused by Streptococcus agalactiae, we have those early onset infections. These occur in neonates that are less than 7 days old. This is due to vertical transmission from the mother to the baby in utero. So what happens is the baby in the first couple of days of life gets pneumonia or meningitis with Strep agalactiae in the bloodstream. This is a highly fatal infection. Late onset group B strep infections occurs between 1 week and 3 months after birth. Usually this is a meningitis that develops. The mortality rate is not as high as the early onset infection, but anytime someone gets meningitis, that could be a very severe infection. So it still is associated with mortality. In adults, Strep agalactiae causes invasive infections more commonly in immunosuppressed individuals or those that have underlying chronic diseases. Sometimes it has been associated with a person that was healthy who just had a child and then can get an invasive infection because the organism went from the urogenital tract and got into the bloodstream. In the laboratory, group B strep or Streptococcus agalactiae is a grayish white, somewhat mucoid, creamy organism with a very narrow zone of beta hemolysis. So if you had Streptococcus pyogenes or group A strep on a blood agar plate next to Streptococcus agalactiae or group B strep, the strep pyogenes would look much more beta hemolytic. It has a larger zone of hemolysis. So with group B strep or strep pyogenes, strep agalactiae, you really need to hold that blood agar plate up to the light, and sometimes the zone of hemolysis is so narrow you can barely see it. To diagnose group B strep, you of course want to do your catalase test to differentiate your staph from your strep. And Strep agalactiae is bacitracin resistant, unlike Strep pyogenes, which is bacitracin susceptible. Other tests are Strep agalactiae is bile esculin negative. It doesn't grow in 6.5% NaCl, and it's CAMP test positive. And this is a picture of the CAMP test. How you perform the CAMP test is you take a blood agar plate, you put a line on that plate of Staphylococcus aureus. Perpendicular to the Staphylococcus aureus, you put a line of your strep, not completely touching the staph line, but very close to it. What will happen is you'll get enhanced hemolysis between the Strep agalactiae and the Staphylococcus aureus, and it will form an arrow right at that junction between the Staph aureus and the Strep agalactiae. Here's an identification flow chart for your beta hemolytic Streptococcus. So you have an organism that was just identified from a patient. You know it's a gram-positive coccus. you have already done a catalase reaction and it's catalase negative. So you know it's not a staph, you're looking for a strep. You look on your blood agar plate and you see that there's beta hemolysis. So how you would identify your beta hemolytic streptococci is you would do a bacitracin, drop a bacitracin disc. If it's susceptible, in other words, if there's a ring around the bacitracin disc, it would be considered group A Streptococcus or Streptococcus pyogenes. If it's resistant to bacitracin, the organism grows right up against the disc, then you would want to do a CAMP test. If it is positive for the CAMP, you get that arrow reaction between the Staph aureus and the strep, it would be considered a group B Streptococcus or Streptococcus agalactiae. If it's negative for CAMP, it could be one of the other Streptococci, and we will talk about those in the upcoming lectures. So we're going to move on now to part 3, where we're going to talk about some of the other Streptococcus species.